Good morning, doctor. I think you are in a new. Yes, now it is audible.
Yeah. Hi, yes.
Hi,
We
hi.
still got 5 minutes, right?
Yes,
Good morning, Dr. Sheikh. Um,
my colleague Dr. Asini Chauhan is also online. So we'll be starting it by 12. We have 5 more minutes for the participant to join.
yeah, so we'll just wait. So,
We'll wait for 5 more minutes. So
okay.
then we can. Okay. So, Dr. Aswini, are you with me?
Yes, I'm here.
So it is actually the time now to go online. So our respected speaker, Dr. Afzal Sheikh is here. Even the second speaker, Dr. Richi is also here. I'll request you to
Perfect.
kindly take it from here.
Yeah. Okay, good afternoon, everyone. So, welcome to the second day of the webinar on drug discovery. And so, the first speaker of today's talk is Dr. Afzal Basha Sheikh, and he's working as an associate professor at Vignan Pharmacy College, Guntur, Andhra Pradesh. He obtained his M Pharma and PhD from Andhra University, Visakhapatnam, and he went to the University of East London, UK, for his postdoctoral research. And Dr. Sheikh has more than 12 years of teaching and research experience, and he has published more than 30 research articles, and he is on board of editorial member and reviewer of several pharmacy and allied health science journals, and he is recipient of several awards like UGC Maulana Azad National Fellowship, Commonwealth Professional Fellowship, which he used to go for his postdoctoral research in London, and Professor S. Rangasamy Gold Medal, ABAP Young Scientist Award, and IB Hub Super 30 Teacher Award. So lots of award he has been given. And Dr. Sheikh's main research area is designing and synthesis of novel heterocyclic compounds with potential antimicrobial, anti-tubercle, and anti-cancer activities. And welcome, Professor Sheikh. We are pleased to welcome you onto this webinar, and we look forward to your talk. Dr. Sheikh, please take over.
Thank you so much, Dr. Ashwin Chauhan, for the brief overview. And I would like to thank Dr. Pratap Acharya for inviting me for this session. And I would like to start my session. Shall I proceed?
Yes, please.
Yeah. Just a second.
Just select the present now button.
Yeah. That's what I'm saying. I am unable to see my PowerPoint here. Just a second, please.
Sure. You can just open the PowerPoint and then
it's open already. That's what I'm looking for. It's open already. Yes. Thank you so much for the brief introduction. And today I am going to explain a part of our research on heterocyclic chemistry. And one important heterocyclic ring that is of interest for our is 1,5-benzothiazepine scaffold. And many heterocyclic, if you observe, heterocyclic chemistry plays a crucial role in the designing and development of the novel drug molecules or drug-like candidates because of their greater potentiality with interaction with the multitude of receptors or the target proteins, and they have a privileged nature because they can interact effectively, and they will also have better pharmacokinetic and pharmacodynamic properties. And if you see the recent statistics in 2019, out of the 26 approved drugs by the US FDA, 22 drugs contain heterocyclic. So it itself explains the importance how heterocyclic rings are essential for the design and development of a novel class of drugs. So coming to our topic of interest, that is exploring novel 1,5-benzothiazepine derivatives as prospective anti-cancer agents by inhibiting epidermal growth factor receptor tyrosine kinase, right? And before going to the details of our study, let's have a brief introduction of cancer. And if you see, just a second, yeah. Yes. And cancer is, all of you know that it's the uncontrolled growth of the cells, and the cells that have been divided will invade and spread to other sites of the body through metastasis, and it may also induce the cancer at the other sites, leading to complications of the overall physiological system. So it is one of the important complications which is responsible for the death worldwide. And according to the recent World Health Organization statistics, cancer figures the second leading cause of morbidity and mortality in 2018. So it itself indicates that development or research in the area of cancer is crucial, because it is one of the leading causes of the death for most of the people throughout the world. And there are 14 million new cases rised in 2018 itself, and 9.96 cancer-related deaths have been occurred in 2018. And the expectation is that this particular cancer cases may rise by about 70% over the next 2 decades. And as all of you know, we are having a different lifestyle now. We are having food habits have been altered, and the lifestyle has been altered. So there is greater chances that we are much prone to cancer. Hence, lifestyle modification is crucial for overcoming this cancer, as well as development of better drugs is also an essential area which we need to answer as a scientist, right? And coming to the gender differences and the occurrence of the different cancer, particularly in the females, the breast cancer, colorectal cancer, lung cancer, cervical cancer, and stomach cancer are more commonly seen. Whereas in the males, the prostate cancer, lung cancer, colorectal cancer, stomach and liver cancer are seen. And here the common point is, in our study, we have performed our activity of our 1,5-benzodiazepines on the prostate, colorectal, breast cancer cell lines. And if we observe the statistics, the breast cancer, where the deaths with the breast cancer is 2.09 millions, and the colorectal cancer is 1.80 millions, and the prostate cancer is 1.28 millions. So all these statistics indicates that the focus on the development of the chemotherapeutic agents is crucial. So there will be a question why we have many drugs for the treatment of the cancer, ranging from the small molecules to the monoclonal antibodies. But despite of availability of the large number of the drugs, we are unable to reduce the statistics. So some of the problems with the existing chemotherapeutic agents is the gaps that have been found are, one common problem with most of the anti-cancer agents is obnoxious side effects. The side effects are limiting or hampering the treatment of the cancer, because if the drug is taken for treatment of the cancer, the patient may suffer with some other side effects. One common side effect, as all of you know, there is a excess of vomiting as well as nausea due to the access of the most of the anti-cancer agents to the chemoreceptor trigger zone, as well as alopecia, as well as there is neutropenia. All these are some of the common problems with most of the chemotherapeutic agents. And the anti-cancer agents not only target the cancer cells, they also interact with the other cells of the body, and leading to non-selectivity, a crucial problem. And the most important problem with the chemotherapeutic agents is multi-drug resistance. This is one of the main hot area where all the people who are working in oncology needs to be focused. The multi-drug resistance is literally making down all the drugs that have been developed, so that we need for some alternatives. So all these are some of the limitations or the gaps that are existing with the present therapeutic agents for the treatment of cancer. And our target here is the benzodiazepine. I will go with the reasons. And the 1,5-benzodiazepines is the basic structure of the 1,5-benzodiazepine molecule, and it will exhibit the tautomerism. And these are the two tautomeric forms of 1,5-benzodiazepine. That is 2,3,4,5 tetrahydro 1,5-benzodiazepine, as well as 2,3-dihydro 1,5-benzodiazepine. And this form of the benzodiazepine is of interest because based on the literature, most of the molecules having such type of scaffold are more active compared to this molecule. And why we have selected 1,5-benzodiazepines? Because this 1,5-benzodiazepine scaffold is not new to our body. Our body, in the sense, already the 1,5-benzodiazepine scaffold has been utilized by variety of the patients because it is present in number of the drugs like the drugs that are particularly used to treat the cardiovascular problems as well as the CNS problems like the diltiazem, clintiazem. Both these are used as the calcium channel blockers. This is one of the important property of the reason why we have selected 1,5-benzodiazepines because I told that multi-drug resistance is a problem, right? And one of the common mechanism by which the cancer cells will exhibit multi-drug resistance is through efflux of the drug molecule. Because the efflux pumps are like P-glycoprotein efflux pumps, they can be inhibited by the molecules which contain the benzodiazepine scaffold, right? Already it was proved that the diltiazem molecule as well as other calcium channel blockers like verapamil, they can inhibit the P-glycoprotein of the cancer cells, and they can prevent, whenever they are added along with the anti-cancer reagents, they can prevent the multi-drug resistance, the combination. So, as it's not a new molecule, it is present in many of the other CNS drugs like clozapine, thiazide, as well as the quetiapine. In the cardiovascular drugs, these scaffold can, when enter into the body, it can accommodate well because the pharmacokinetics of this molecule or this scaffold are better understood. So it means that if this scaffold can be used as a drug for designing the drugs. So it's a privileged structure in the designing of the new drugs. So this is one point why we have selected benzodiazepines, because the access of the benzodiazepines to the body is well, and it may have very good pharmacokinetic properties, right? And not only that, literature also suggests that there are many molecules which are benzodiazepine derivatives which are with potential anti-cancer activity. This is some of the literature where we have dwelled into, where there are many 1,5-benzodiazepine scaffolds with potential anti-cancer property. So considering that point that they have the ability to access the body well, as well as the potential anti-cancer activity of some of the existing literature, suggestions from the existing literature represents that definitely we can prepare these molecules and we can move forward to develop new lead molecules against the cancer, right? And the basic idea or the design of the molecule here is they contain the benzodiazepine scaffold, which we are considering the thiazepine ring as A, and there is a ring B, and there is ring C, and we kept this ring C constant. There is an hydrophobic ring C that is which contain isobutyl phenyl ring, which was kept constant, and we have altered the ring B and we have tested these compounds. In ring B, what we have done is we have used different aromatic rings containing different electron releasing or the electron withdrawing groups, as well as we have also incorporated heteroaromatic rings, and we have studied their anti-cancer effect on the 3 cancer cell lines, as well as we have also performed its activity on what we say the epidermal growth factor receptor tyrosine kinase. So this is the thing. The basis for design of benzodiazepines containing 3 ring structures can be attributed due to following reasons. And if you see this, you can see a complex lipophilic nature of this molecule because it contains a T3 phenyl rings. Say, for example, if you take the ring B, a simple phenyl ring, this is one phenyl, this is phenyl. And overall, the molecule has the required lipophilicity so that it can effectively interact with the, it can cross the barriers, and it can enter into the cancerous cells. The compounds with structural features will have better lipophilicity, which aids in the better transfer of these molecules into cancerous cells by the crossing the lipophilic barriers. And as it has multifunctional, here you can see 3 different types of the rings. Yeah, so it is a multifunctional in nature. Due to its multifunctional nature, automatically there is a greater chances of interacting of this molecule with the existing cellular targets, that is cancer cellular targets or the proteins, or sometimes we can also probe, whenever we probe its mechanism, there is chances of identifying a new target where the benzodiazepine can show their action, right? And the benzodiazepines, as I told previously, will inhibit the efflux protein and may also show dual action as anti-cancer agent, as well as P-glycoprotein inhibitor to overcome multi-drug resistance in cancer. Right? So this is the reference work which has highlighted the importance of benzodiazepines as a P-glycoprotein inhibitor to overcome the multi-drug resistance. And this is the synthetic scheme. The target compounds are labeled as B1 to B20. The target compounds have been synthesized starting with isobutyl estophenone, which is condensed with a variety of aromatic aldehydes by a Claisen-Smith condensation reaction. And this scaffold is generally called as chalcone. And this chalcone moiety, which contains the alpha-beta unsaturated carbonyl system, is highly reactive, and it is helpful for the synthesis of variety of heterocyclic rings. And this is one beautiful moiety, or the functionality, or a synthon, chemical synthon, that will help us to convert into a variety of heterocyclic rings. Not only the benzodiazepines, we can synthesize a variety of molecules ranging from 5, 6, as well as 7-membered rings. You can synthesize isoxazoles, you can synthesize pyrazolines, you can synthesize pyrazoles, as well as you can also synthesize pyrimidines, as well as you can synthesize pyrons, and you can synthesize benzodiazepines and benzothiazepines. We already written some interesting review about its reactivity, biological and synthetic potentiality of chalcones. There is one important, interesting review we have published. You can refer that for further understanding the reactivity of this. And this, when reacted with this molecule binucleophilic substituent group, that is 2-amino 2-mercaptothiophenol, it results in the formation of the target benzodiazepine scaffold. And for this, we have used simply methanol and piperidine. Piperidine is used as a basic catalyst. Upon refluxing it for 8 hours, around 5-8 hours, we got the target compound, which will be separated as a white paper-like substance. Right, and I told that both these are constant. The isobutyl phenyl ring as well as the benzodiazepine scaffold are constant, and we have altered the nature of the ring B, and we have used a variety of aldehydes here, which has been continued in the ultimate benzodiazepine scaffold, and we have used a range of aldehydes containing different types of electron withdrawing as well as electron releasing groups, and we have also used some isosteric replacements. If you observe this, up to compounds B14, they contain chloro, methyl, dichloro, fluoro, as well as the bromo, hydroxy, and dimethoxy, trimethoxy, nitro. All these are the different electron releasing and electron withdrawing substituents. But we also tried replacing these part of the molecule with some of the isosters of these phenyl rings. Like we have incorporated the 2-pyridinyl, 3-pyridinyl, as well as the 4-pyridinyl, and this is the 6-membered rings, as well as we also tried to incorporate the compounds containing 2-pyrolyl, 2-tienyl, and bromofuryl systems. So we have analyzed the activity. After poor activity of this, we have synthesized the isosters of this, and then we have tested the anti-cancer activity of these isosters. Yes, and we have characterized all these compounds by proton NMR, IR spectroscopy. IR spectroscopy was vital in identifying this particular carbon double bond end, because I told that there is chances of formation of either this type of benzodiazepine or this type of benzodiazepine, but the IR spectroscopy has helped us for differentiating these two compounds. There you can we haven't observed any sort of even I haven't represented any IR spectrum here. We have not seen any NH absorption band in the IR spectrum, but we got an intense carbon double bond nitrogen absorption band, representing that our target molecules contain this scaffold but not this. Yes. Yeah. And if you observe this NMR, in the NMR we got we have 3 carbon atoms here, the second one, third one, as well as fourth one. and this is forming the benzodiazepine scaffold by interacting with the 2-amino-thiophenol. And the second carbon atom, we have observed a doublet of doublets at 5.09, and there is also a doublet of doublets for at 3.56 for the there are two protons, that is carbon 3 hydrogen 3a as well as a 3b. It is something like ABX system, and it will couples with this particular proton. Due to this coupling of the proton on the second carbon atom as well as the coupling of the proton of the same carbon atom, which is stereochemically different, we are able to see a doublet of doublets at 3.56, as well as we have also seen a doublet of triplet of C3H3b, which is stereochemical interacting with this particular proton of C3HA proton as well as C2H proton, and these are the J values representing the conformation. So these three are crucial in confirming that our target molecule has been generated. If these are absent in the spectrum, these area, then we can come to a conclusion that the target compound has not found. And there are also other peaks of this, that is, this represents the other 12 aromatic hydrogens of the scaffold, as well as the alkyl group. There is a isobutyl group, hydrophobic isobutyl group, which has given the corresponding peaks in the NMR spectrum, right? And not only the proton NMR, we also have other important carbon-13 NMR spectrum. The carbon-13 NMR spectrum has given the characteristic peak at 59.10, as well as there are also peaks corresponding to 40.10, the third carbon atom, as well as the fourth carbon atom. And there are also other carbon atoms which are represented here. Ultimately, we are able to derive the compound. And we have also scanned all the compounds for their mass spectrum. And it was taken in a positive mode through positive electron spray ionization technique. And due to its positive mode, actual molecular weight of this compound was 405, as it was taken in positive mode, where this nitrogen can take a proton, because in positive mode we will have what we say acidic medium. In the acidic medium, the nitrogen, which is basic, can take up a proton, so that the molecular weight will get somewhere extra around 406. And we also have a chlorine atom in this molecule. Due to presence of the chlorine atom in the molecule, we have also observed another peak, that is M+2 peak at 408 with the M/Z value of 408, right? So all these spectra has confirmed that the molecule has been generated. And similarly, not only I have taken one example here just for our understanding, all the compounds have given the similar pattern of the spectrum, and they have given their respective peaks depending upon, as we have discussed, everything is constant here. There is change in this. Depending upon the changes on that, we have got the respective spectrums. And we performed anti-cancer activity by the well-known and still used, even though it was developed in 1983. And there are minor modifications of this method, that is known as Moskman's MTTSA, upon 3 cancer cell lines. One is the colon cancer cell line, the other is breast cancer cell line, and prostate cancer cell line. Considering the statistics that taking into consideration that both the male and female are affected by colon cancer, as well as the prostate cancer is specific for males and the breast cancer is specific for females. But the common point is colon cancer. And these 3 common cancers are very common in both overall, both males and females are affected by colon cancer. And specifically, the females are having breast as well as the males are having the prostate cancer. Considering that statistics, we have selected these cell lines. And also the report suggested that benzodiazepines also has a very good activity on these cell lines. Previously, already I have shown some of the structures where we have took the basics for understanding the activity of 1,5-benzodiazepines. And these are the results of anti-cancer activity on the different cell lines. And we started with chlorinated as well as electron releasing, electron withdrawing groups. as well as we have also replaced, I told, from B15 onwards, we have replaced them with pyridinyl systems, as well as pyrrole, thiophene, as well as furan systems. And whenever we tested first these compounds up to B14, we got less activity than that of the standard methotrexate. As there was less activity, we thought of replacing this with some heteroaromatic rings. So we have started with the pyridinyl systems, but the pyridinyl systems are even devastating. The activity was even less compared to that of the compounds which contain. Particularly whenever the phenyl ring is present, the phenyl ring substituted with electron releasing and electron withdrawing groups. Whenever it has electron withdrawing groups like halogens, the chlorine as well as the fluorine atoms, that is, they have some interesting activity, but it is not less than that of the standard methotrexate. So we tried with the heteroaromatic rings of 6-membered size, but unfortunately the activity has been reduced. And the activity of the electron withdrawing groups, electron releasing groups like trimethoxy as well as dimethoxy, it is even less. And there are some cases where there is no more activity. There is no activity with the electron releasing groups. And after replacing this 6-membered scaffolds with the 5-membered scaffolds, suddenly the activity has been interestingly increased, right? That's why we started with this molecule. There is an increase in the activity, but the activity is reduced in this case. However, whenever we incorporated electron withdrawing group on the 5-membered ring, the activity was amazing, and it was even more than that of the methotrexate on all the 3 cancer cell lines. So we come to a conclusion that this particular portion containing 5-membered ring is crucial for the activity, and when it is present along with an electron withdrawing group, definitely we can get a potential activity. And we have got activity somewhere around 2 as well as the 4 microgram per ml from, on the different cancer cell lines, breast cancer as well as the colorectal cancer and the prostate cancer cell lines. And these molecules also possess somewhat interesting activity, but it was less than that of the methotrexate, but they have close activity to the methotrexate. So we thought these are also the potential candidates where we can do modification of this ring by incorporating electron withdrawing groups, as it has given a promising activity. So then the potency of these molecules can also be altered, which we are trying to do as of our next study. And then after performing the anti-cancer activity, we need to deconvolute the target. Deconvolute the target means we need to identify the exact target where they are interacting to show their activity. So in order to satisfy our results, we have performed its activity on the benzodiazepines activity on epidermal growth factor receptor. Why we have selected epidermal growth factor receptor tyrosine kinase is there is some important criteria for selecting this target for our study, because epidermal growth factor receptor tyrosine kinase is one type of tyrosine kinases which is very much important in inducing the cancer. Whenever it is activated, there is a vast rise in the chances of cancer. And there are variety of cancers where the EGFR is highly expressed. That includes the gliomas, the cancers of colon, breast, lung, pancreas, kidney, bladder, and ovary cancer. And here, if you observe the results of our anti-cancer study on the cell lines, and the anti-cancer activity was good among the colon as well as the breast cancer cell lines. The colon cell lines, that is ST29, as well as breast cancer cell line MCF7, if you observe, it has interesting potential activity compared to that of the prostate cancer. Breast cancer cell lines. Overall, if you observe the breast cancer cell lines, the prostate cancer cell line is having 15 microgram per ml, and here also the prostate cancer cell line is having 80 microgram per ml. Overall, if you compare these 3 compounds, we have got compounds with good activity on the colon as well as the breast cancer cell lines. As the colon and breast cancer cell lines are expressed with excess of the epidermal growth factor receptor tyrosine kinase, we thought that it may be a potential target for the interaction of these molecules, the benzodiazepines. Right. One important point here is we have also tested our compounds on the normal cancer, normal cell lines, that is the normal healthy human cell lines, LO2, and as their activity was greater than 40 microgram per ml, and it can be considered as safe, and none of these compounds are toxic, toxic, so they can be considered as safe molecules. And considering the potential activity of these new synthesized 1,5-benzodiazepines on colon and breast cancer cell lines, as well as overexpression of EGFR, epidermal growth factor receptor tyrosine kinase, in these two types of cancers, and structural resemblance of 1,5-benzodiazepines with existing epidermal growth factor receptor tyrosine kinase inhibitors, we further evaluated or screened these compounds against EGFR, epidermal growth factor tyrosine kinase enzyme. So I told that not only this point, that is expression and good activity on these cancer cell lines, and other important point is structural resemblance. If you see the marketed epidermal growth factor receptor tyrosine kinase inhibitors like erlotinib, afatinib, and gefitinib, and they have common bicyclic ring here and an hydrophobic substituent like phenyl ring here. So considering this framework, what we thought is our molecule contains a bicyclic framework with an hydrophobic group which is constant without altering. And we also we have altered ring B, and as this particular group has greater impact on the activity, we thought that considering these 2 points, we thought that if we test the compounds similar to that of these compounds, definitely we may get some good result, and we can deconvolute the target of this novel benzodiazepine derivatives. So we performed the EGFR tyrosine kinase activity by following the protocol established by Cormi et al. Group of Italy, and which was published in Bioorganic and Medicinal Chemistry Letters. We have adopted the same procedure, and these are the results. And amazingly, the point here is, in the previous case, I told that the compounds which has the electron withdrawing halogen atoms as well as the 5-membered rings have got excellent anti-cancer activity on the cell lines. So the same scenario has reflected in this, reflected in the epidermal growth factor receptor tyrosine kinase inhibitor activity, suggesting that this will be the most important target for this class of compounds. And even though the activities was less than that of the erlotinib, the standard drug which we use for this study, erlotinib has a inhibition of percentage inhibition of 71.2%. Our molecule, that is B20, which was more potent in the cell line testing, was also active, was the most potent, and the scenario has continued with the even with the halogenated compounds contained on the B ring. So this represents that definitely benzodiazepines has ability to interact with epidermal growth factor receptor tyrosine kinases. And see, if you see the same structures which has reflected here. Here we have performed the activity at 10 micromolar concentration, and the standard used was erlotinib at 0.001 micromolar concentration. And again, this molecule has excellent activity, and again, structural modification of this molecule will definitely leads to increase in the activity similar to that. And one important advantage of these molecules will be that in the case of erlotinib or the other molecules, they have the resistance. There is a ABC transporter of the cancer cells, and the ABC transporter of the cancerous cells will efflux this erlotinib and afatinib, through which they have some resistance. That is one of the limitation of these drugs. And as I told previously, benzodiazepine molecules has greater propensity to inhibit that ABC, which is a glycoprotein ABC transporter. Definitely, we are expecting. We need to test further. We are in the process of studying further these molecules. So definitely there will be a chances of overcoming the resistance. As I told, the benzodiazepines may have dual activity. One is the anti-cancer activity by interacting with the epidermal growth factor receptor tyrosine kinase, and the other one is they may also inhibit this calcium channel, that is ABC transporter, a type of calcium channel. So, but it has to be studied. And we felt one important reason for the activity, if we compare with that of the erlotinib, the activity of these molecules is less. The activity of this molecule at 0.0001 micromole concentration is 71.2, but we have tested these molecules at 10 micromole. That means even at 10 micromoles, this is unable to interact as effectively as erlotinib. That represents that there should be some structural modification of these moieties, which may be helpful. So we have observed that this part of the molecule is okay because it is having the benzodiazepine scaffold, a bicyclic ring like that of this, as well as a hydrophobic group. But this part of the moiety has not been explored. So we are studying further by exploring the phenyl ring. Now we are focusing the derivatives which has a substituent something like this on this benzodiazepine ring, and definitely we are expecting very good results of after modification of this, so that it can have potency equivalent with that of these existing drugs, or sometimes wonderfully we may also get more activity. So our now hot area or the hot ring which we are concentrating now is this particular scaffold without altering this. And sometimes we can also alter these rings, and we get closer to the activity of erlotinib. That should also be tested by incorporating different 5-membered rings with electron withdrawing substance. So these two are the areas which we are trying to explore further without altering this. In order to understand its properties, we have also performed a small computational study employing the phase of Schrodinger software to determine their essential groups that might be possibly interact with that. We have not performed the molecular docking studies. That is, we are also initiating the molecular docking studies further, so that priorly we have performed the computational studies before tyrosine kinase activity. Then only after that we have performed the tyrosine kinase activity. And first we have just performed this activity, that is pharmacophore based models. We have developed the pharmacophore based models, and we have developed one active model out of this, where it has 5 important portions of the molecule are essential for the activity. And the model what we have developed is a 5-point model, that is AHHRR model, which has the different chemical features. One is the hydrogen bond acceptor. A is a hydrogen bond acceptor, as well as we have the hydrophobic groups, that is H5 as well as H4, as well as there are ring residues or the aromatic rings, R7 as well as R6, right? So this is the basic, after aligning all the 20 benzodiazepines synthesized, we have got a common 5-point model using the phase of Schrodinger and the 5-point model. And interestingly, the molecule B20, if you observe, it has an excellent property similar to that of that. And if you, this is the B20, even after, this is the best pharmacophore hypothesis, which have been extracted out of our study. And we found the ring residue here, and there is an hydrogen bond acceptor as well as the hydrogen bond donors, hydrophobic groups. And these are the 2 ring residues. And this molecule has aligned exactly according to requirements of the best pharmacophore model. And BT20 has given very good result in the cell line assay, as well as in the epidermal growth factor receptor tyrosine kinase inhibition assay, as well as in our pharmacophore models. So definitely this will be a promising candidate further. So we are exploring this part of the ring, particularly the benzene ring of the benzodiazepine scaffold, in order to further understand. So these are the common pharmacophore model hypothesis, pharmacophore hypothesis, AHHRR162, where you can see the different distances and angles of this particular model. So if the molecule, out of the 20 benzodiazepines, the benzodiazepine molecule which has more exact angles as well as distances along like this will fit exactly into that model, and that will be separated as a best candidate developed from the pharmacophore hypothesis, right? So with this, I would like to conclude. We have synthesized a variety of pharmacophores in good yields, and we have evaluated them by spectral studies, and we have performed the anti-cancer activity as well as anti-EGFR activity. And there was some promising activity in anti-cancer activity, as well as they also have a good tyrosine kinase inhibitor activity. Further structural modification are needed. And as said, we have utilized a variety of aldehydes in order to synthesize containing electron withdrawing as well as electron releasing groups. And we saw that electron withdrawing groups have contributed excellent for the activity. And in specific, whenever the electron withdrawing group is in association with a 5-membered heterocyclic ring like furan, it has a good activity. And we are trying to attempt to introduce substance on the 4-isobutyl portion as well as on the benzodiazepine portion. And computational studies also has highlighted the important structural requirements and given good insights and clues for the synthesis of new benzodiazepines further. And we conclude simply that these novel benzodiazepines will be useful as lead molecules for further development of new cancer therapies, particularly against the colon as well as the breast cancer. And future outlook is as we are trying to modify the molecules, the most important molecules, and we need to test the anti-cancer activity in animal models too, as we have performed only cell-based assays as well as enzyme-based assays. And one important limitation of benzodiazepines is they can interact with the CNS, and there is possibility of CNS and CVS problems, because we should also screen that in vivo in the animal models if they are having any effect on the CM brain as well as the heart or the cardiovascular problems. So we are working in that direction. We are first trying to improve the activity so that they can have activity more closer to that of the epidermal growth factor receptor tyrosine kinase inhibitors like erlotinib. And once if they are competing with that, then we will go with this testing, anti-cancer testing in the animal models, as well as we'll also test for the toxicities if there are any on the CNS as well as the CVS. And this work has been published in anti-cancer agents in medicinal chemistry. And these are some of the students who are working with me. They have not involved in this project, but these are the group of students who are working under my guidance for their undergraduate as well as a postgraduate thesis. And they are very interested and enthusiastic people working for designing and synthesizing a variety of heterocyclic scaffolds. And they are much interested in heterocyclic chemistry. There are some students who are Bachelor of Pharmacy students. These two students are Bachelor of Pharmacy students in our college. And later, because of their interest, and this guy has worked along with me for his Bachelor of Pharmacy thesis. And because of their interest and passion in heterocyclic chemistry, they have again rejoined in our college for their pharmacy. And they are working. They have completed their thesis recently, and they have also entered into the job. And due to Corona crisis, they are they need to go back to the job after some 1 or 1.5 months. And this student has rejoined in our college as a faculty, and these are some of the other students who are still working under my supervision. And he is Dr. Mukleswar Rahman, one of our collaborator and my mentor during my postdoctoral research. And he is Dr. Kandarpa Kumar Saikia, who is also working along. He is one of our collaborators. Now we are working associately, and he will perform the biological activity. He is from University of Guwahati at the Department of Biotechnology. And this is the lab where I worked in the UK for my postdoctoral research. And he is my guru, Professor Ejala Rajendra Prasad Garu. one of the pioneer in Chalcone as well as heterocyclic chemistry. There are a number of citations, many number of citations for him, and he is the base for our development, who has enhanced our enthusiasm towards the heterocyclic chemistry. He has given many PhDs, somewhere around 60 PhDs and 10 postdocs. He is Dr. Muklesh Rahman, as already seen. And I would like to, there are also many collaborators due to time constraints and other factors. I was unable to keep their photographs. And one important collaborator is Dr. B.V. Subramanya Lokesh, who is based in Malaysia, who will do computational work. We have collaborations and we have published some papers. And I have also collaborations with Dr. Richie Bandare, who is a speaker today at Ajman University, College of Pharmacy. These are some of the other collaborators. And I would like to extend my thanks to University of East London, as well as the Commonwealth Scholarship Commission, as well as I would like to thank my base university, Andhra University, which has nurtured me, and my present organization, Vignan Pharmacy College. And I am ready for questions now. Dr. Acharya.
Thank you, Dr. Afzal. I'll ask my colleague, Dr. Asuri Chavan, to take on, please.
Yes.
Thank you, Dr. Sheikh, for your Dr. Afzal. It was a fantastic talk on drug designing.
Thank you so much.
EGRF molecule that you are trying to make. So we are open for the question. There's no question. If I have a question, being a biologist, for me, all these, pharma things are very intriguing. I get, excited and, I want to jump onto these molecules always, So how we can work on these. And so my question is, like, you looked at EG, you're looking at this epidermal growth factor, right? RF, which is EGRF. And it's a family of 4 different groups, if I remember correctly.
Yeah, exactly.
So have you looked into those, because these are very structurally similar, have you looked into those factors? Because epidermal growth factors are very important. As you said, the central nervous system is directly affected if there is a, kind of a defect in any kinase activity there. These are tyrosine kinases. And this intracellular signaling is very important. You get Alzheimer and many things if these are perturbed. So have you looked at these 3 other families, the representative?
No, we haven't studied the other families, but we are much focused on the epidermal growth factor first. We haven't studied the other families.
Yeah, because in cancer, EFGR is going to go up dramatically. The expression. That's why you targeted. But other factors, if they are perturbed, they might affect, the growth of epidermal.
Yeah. That is our future study. We are looking into the animal model. First, we are trying to increase the potency of these compounds, because they are not as potent as that of the existing marketed formulations. So here what we are trying is, as a medicinal chemist, first I am trying to increase their potency in the cellular models as well as the enzymatic models. If once the most potent compound has been derived, then we will switch on to the animal models as well as the toxicity on the animal models. So then we can get a better understanding. As of now, we have not performed on the CNS activities or the other activities. We are trying to explore that area also.
Dr. Afzal, I have few queries.
Yes, sir.
So have you used any other electron withdrawing groups like the trifluoromethyl groups?
No, we haven't used it. It also because as bromo derivative has given excellent activity, we are also in the plan of using trifluoromethyl, trifluoromethoxy, as well as trifluorosulfide. These are the 3 important.
Yes. I feel these halogens can be major player in the biological activity.
Yes.
Because recently we also did some anti-cancer activity and we saw that this halogenated derivatives are good, but methoxy, as you said, methoxy derivatives are useless. They are not giving any activity. Second, I saw your compound structures where you have attached the ring B in the benzothiazepine ring. ring B is attached in the benzothiazepine ring. If you see
Yes.
the Zeptinib or Erlotinib, your this ring, these attachments are on the phenyl ring. So you can think that you can attach the ring B into the phenyl ring rather than putting it onto the benzothiazepine ring. Have you thought of that? Ring B. Your ring B.
Yes.
Your ring B is attached to the second position of the benzothiazepine, right?
Yes.
So instead of putting it into the benzothiazepine ring, you can just shift the ring B onto the phenyl ring.
Yeah.
Benzene.
That is an interesting point. That's an interesting point.
Because if you see the
If you
drugs,
observe the structures of EGF or tyrosine kinase inhibitors, actually the scaffold is there.
yes,
And the other
yes.
part of the molecule. Exactly, you are 100% right. That is what we are trying. Because what my intention is, we need to keep this portion as such and to incorporate a new phenyl ring so that we can develop a new pharmacophore.
But I
That
feel
is
if you
right.
can shift that, it will be similar. It will be having
You are
similar
absolutely
structure
right.
and can give you good result.
That is also one important. We can completely remove that portion and we can incorporate a phenyl ring in that area. So there is greater chances of getting activity also. You are absolutely right. That's a nice point too.
Yes, you can just explore those areas. Yes.
Yes, sir.
I
Thank you so much.
I think there is one more question from the audience. You can see the chat box, and Dr. Ashwini will help you.
Yeah, so the question is, can you prepare a benzothiazepine in nanotechnology for chemotherapy?
Can we prepare benzodiazepine
In nanotechnology for chemotherapy?
nanotechnology for chemotherapy? Maybe. Yes, we can. Yes, we can. We can formulate the nano formulations using these molecules. Definitely. Yes. The answer is yes. We can explore that.
Any other question from the audience?
I think there's no more questions, so maybe.
So you please take on the forum.
And so we would like to thank Dr. Sheikh for this interesting talk. You know, the session is going really beautiful. You know, especially I was thinking that, you know, when Pratap asked me to join, I was like, you know, it's all pharmacy, but I'm learning so much. You know, there's so much biology in there. Thank you so much.
Yes. Biology. Without biology, we can't do anything.
So thank you so much for taking the time to this webinar and wonderful talk. Thank you so much. We appreciate it.
Thank you.
I think we would take a leave now until the afternoon session.
Yes, we have a lunch break now.
So we all can join at 1:55 before the next speaker is there. So
1:55 will be fine.
1:55, right?
Yes.
So perfect. Thank you. Thank you so much.
Thank you, Dr. Pratap, for calling me. Thank you so much. Bye bye. All of you have a nice time. Bye bye. Bye bye.
Thank you.